I need to speak on the thing to be there. So we need to speak in the microphone, Rob and Sam. So a couple of logistical things I want to say, and then we start. But we, I think I mentioned in some meetings, but not to, I'm going to keep repeating, we changed the way that we are virtualizing these grand rounds. From before we have a system that allowed for interaction. Now we have a different system that is going to be recording, and people get a website link that Peggy Falkin has sent around, so they can watch this from their computer desk. That, I think, works better. It's more resilient in terms of getting the actual lecture and so on, but it will not allow for communication, so for questions. So if there's people listening to me right now in the computer screens, they want to ask questions to Rob, they can go ahead, text me my cell phone, or call me my cell phone, and I will pass the questions to Robert. The other thing is the people who are here in the room who want to claim CMEs, they need to follow this procedure. The people who are in the computer, wherever you guys are, you also need to do the same mechanism. And lastly, all faculty should also sign when they walk into the room. And if you are listening to this, you should send an email to Peggy that you are listening to this. That's it. Thanks.
All right. And also, if you want to claim the CME credit, you need to, these are only for people who are doing it for CME. You need to text the 9720 to the number on the screen. So hopefully that system's working well. I just tried. I haven't heard back yet. But so for today's Grand Rounds, we're thrilled to have Robert Stevens. Dr. Stevens is an associate professor in almost every department in this hospital. He's an associate professor in neurology, neurosurgery, anesthesiology, and radiology. Is that correct? Got them all? All right. And one of the attending physicians in the NCCU, and very involved with that, and has a strong research interest in effects of traumatic brain injury, and he's going to talk to you about that, and I think it'll be a great talk for all of us. So with that, I'm going to turn it over, and we'll go from there. Thanks a lot, Robert.
So I wanted to thank Dr. Mayer, Dr. Selnick for inviting me here today. At first, when I walked into this room, I was having some second thoughts because I saw it says Department of Gynecology and Obstetrics, and I wasn't quite sure if I was in the right place, but it seems that I am because I see many familiar faces, and in fact, many of the people that work with me on a daily basis in the NCCU taking care of these very fragile, vulnerable patients. So I wanted to talk about something today that is kind of a fashionable topic. And I think it's something that has generated a great deal of excitement, and it really, I think, potentially represents one of the future faces of medicine in terms of how we practice it. And I'm clearly an enthusiast, but if you guys have questions or if you have skepticism, please feel free to interrupt me and ask questions. And I'm hoping that those who are looking through the simulcast are able to hear my voice and also ask questions, perhaps by calling or texting Dr. Selnick. So I should acknowledge that a lot of this work is the result of a task force that I had the privilege of chairing for the Society of Critical Care Medicine. For the past year and a half, we've been meeting on a very regular basis to try to delineate a research agenda regarding precision medicine in critically ill patients. My focus today is going to be on precision medicine as it applies to traumatic brain injury, but I did want to acknowledge significant amount of support that I received from the SCCM in terms of this thinking through these different ideas. So I don't know, how many of you are fans of soccer? I certainly am. And I don't know if you remember the FIFA World Cup just a few years ago, with a match that involved the team from Germany. And it was really a remarkable game, actually. because one of the German players was actually knocked out during the match. I think he received somebody's knee in his head, and he became unconscious, was lying on the field, as shown here, and for more than 10 seconds was really unconscious. And then slowly came around and started sitting up. He was briefly seen by his teammates, by the team doctors, and they deemed that he was fine. And he continued to play for 14 minutes, actually. I don't know if you guys remember this, but for 14 minutes he was staggering about the field, clearly disoriented, not able to coordinate his playing and his walking. And finally, when it was obvious to many, many people that he was not doing well, he was taken off the field, and he was, of course, sent for further testing. Thankfully, this was just a severe concussion, and no lasting consequences, and this player has done very well since. But it just gives you a sense. This is a game that was broadcast to millions, maybe billions of people across the world, and they let him play for 14 minutes after sustaining what was clearly a severe neurological insult. Nobody did anything about it. And I'm just sort of highlighting this anecdote as an example of something which is very, very common. If this could occur in the World Cup in front of billions of people, I can guarantee you that it's happening every single day in soccer matches around the world, and it's happening also in civilian life, it's happening in the military. It's something which is really, really common. And if we think about the epidemiology of TBI, the best guess, and it's a guess because we don't really know what the true magnitude of this problem is, but the best guess from the Center for Disease Control is approximately 2.2 million emergency department visits, about 250, 280,000 people that are hospitalized each year with TBI, 50,000 deaths, and that death rate, the mortality of TBI is declining. But the real question is how many people actually sustain a TBI? And I'm sorry to say that we don't really know, but the best, sort of guesstimate is that about 3 to 4 million per year in the U.S. alone, 1% of the population is involved in some kind of traumatic brain injury. And when I speak about traumatic brain injury, and I'm hoping that this is obvious for all of you, we're talking about not necessarily only a mechanical impact. We're talking about injury to the brain that is sustained by a very sudden acceleration, deceleration, by a rotation, or by a blast, right? So you don't necessarily have to hit a surface to sustain a traumatic brain injury. So 3 to 4 million in the U.S. If we extrapolate on a global scale, and these are estimates again from the World Health Organization, we're talking about maybe 60 to 80 million cases of TBI each year. So this is not a rare condition. And it's notable that between 85 and 87% of TBIs are concussions. And another notable fact, and this is where the epidemiology becomes really challenging, is that between 30 and 50% of TBIs actually are not evaluated medically in the acute setting. Some of these patients eventually come back for persisting symptoms, but the fact is that this is not a condition that immediately leads to medical consultation in many, many cases, 30 to 50% of the time. And so if we compare, for example, in the US, the estimate of 3 million TBIs a year, this is almost twice as many cases as the incidence of cancer. Of course, the mortality associated with cancer is higher, but it just gives you a sense of the magnitude of this problem. And the CDC, again, believes that between 5 and 5.5 million people are living with the sort of chronic disabilities, long-term functional disabilities associated with TBI. This is 1.6% of the population. And again, difficult to estimate entirely, but the costs associated with TBI are in the region of 100 billion in direct and indirect expenditures each year. So given the importance of this problem, you would expect that there would be a huge amount of funding for TBI research. In fact, the funding for TBI is actually quite miserable and abysmal compared to the funding for many other conditions. This is just an example of all the funding for the National Institute for Neurological Disease and Stroke, so not just TBI. Like TBI is just a tiny fraction of this, maybe less than 5%, compared to funding for cancer, National Cancer Institute. And you can see that it's 2.5, 3 times more money is being allocated by the NIH for cancer research than to neurological disorders. And yet, the magnitude, again, of TBI is, which we could consider potentially one of the most, if not the most common neurological disorder is very, very low at this time. Why is it important to fund TBI research? It's important because, as you guys know, TBI is not an isolated event. It typically leads to long-term neurologic, physical, emotional consequences. So many of you are familiar, of course, with the sort of post-concussion syndrome that can be quite disabling. A subset of patients with TBI can develop seizures. There's a known association between TBI and movement disorders like Parkinson's disease. Motor neuron disease, ALS, is linked to traumatic brain injury. We know that traumatic brain injury can set into play a neurodegenerative process which can lead to accelerated dementia. There's this well-defined tauopathy, which is called chronic traumatic encephalopathy, that has been described in retired NFL players and other retired athletes, which seems to have a much delayed onset with development of a neuropsychiatric syndrome, often leading to severe depression and suicide. Many patients with TBI go on to have psychological problems, including anxiety disorders and mood disorders. So this is not a benign condition, and the question is, can we do a better job of evaluating TBI and treating it? This is just one slide as an example of the potential cumulative effect of aging and TBI on the risk of dementia. And this is a cohort of U.S. veterans, and you can see that when you compare those veterans who have no history of TBI with those who sustained a TBI during their time in the service, the ones who did have a TBI have an incrementally higher risk of developing dementia over time. And this has been described, a very similar relationship has been described also, for example, in players of contact sports, soccer players, football players. Those who have sustained one or several TBIs are at higher risk to develop dementia. So one of the biggest problems with TBI is it's not a disease, it's a disorder, it's kind of a syndrome. And it's extremely variable in presentation and also extremely variable in terms of its trajectory, right? So this slide just illustrates this. You have on the Y scale, you have the Glasgow Outcome Scale Extended, which is, you all know, is the functional scale often used to measure outcomes in TBI. And you can see that this cohort followed over time. Each one of these lines represents an individual subject. You can see that there's an extreme amount of variability. You could start, for example, initially with a GOSC of, let's say, 3, and any range of outcomes is possible. There's a huge amount of diversity or heterogeneity in the individual trajectories of TBI patients. And the fact is that we're not very good. In fact, we're quite bad at the outset when the TBI just happens at discriminating which patients will have a favorable trajectory versus a less favorable one. And so the reason I'm bringing this up is because I think that precision medicine, sort of thought of globally, could potentially represent a solution to this problem. In terms of predictive models, prognostic models used in TBI, in moderate and severe TBI, the best known are the impact score and the crash. These are two multivariate models that essentially combine clinical characteristics and characteristics observed on CT scans, so clinical and CT, sometimes some biochemical or physiologic variables as well. And these models, are currently the state of the art. These are the sort of industry standard in terms of clinical research in TBI. The fact is, though, that they do very poorly. So if we wanted to measure how well they discriminate between favorable and unfavorable trajectories using an ROC or analysis, the area under the ROC curve that we're getting is maybe 70%, 80%, which means that 20, 30% of the time when we use these multivariable models, we actually are completely wrong. We are not able to accurately predict or discriminate between outcome categories. So clearly we need to think about better ways to do this. This is also a depressing slide because it details a number of very well-known, large-scale, multi-center, million-dollar randomized controlled trials looking at different interventions, neuroprotection, hypothermia, to potentially ameliorate the outcome in moderate and severe TBI. This is research carried out over 10 years, and there's many, many other studies that I could show you. The fact of the matter is that there is actually no effective biologically targeted therapy for TBI. There is no treatment for this condition, right? So we're talking about something that affects maybe 1% of the population. We're talking about high potential for long-term neurologic and psychological and functional and problems. And yet, there is no treatment, right? We provide supportive care. We are quite good, for example, in the intensive care unit, treat brain swelling. We can do a lot of different things. We can resuscitate. But really, we really don't know how to fix the traumatized brain. And I think this is a major challenge for us. Which brings me to this question of precision medicine. And, what is precision medicine? I think many of you have already thought about this and read about it. But what we're talking about essentially is data. We're talking about integrating multiple levels of data, molecular data, imaging data, phenotype data, and using these in sort of iterative computational models to arrive at a better classification of the disease. Another way to sort of state this maybe in simpler terms is, precision medicine is really to ensure that the right treatment is delivered to the right patient at the right time. And, I just want to spend a few minutes on this slide because it kind of details how we've arrived at this stage now in precision medicine. So, we're going to work from this box and go up here and then over there. But essentially what we have here on the sort of vertical scale is interventions or detection or diagnoses that are made on an individual scale. And then here, on this part of the vertical scale, we have population-based interventions or measurements. And then here, on the sort of horizontal scale, we have low-dimensional data, and on the other extreme, we have high-dimensional data or precision medicine. And so, in a way, you can say that the medicine as it is practiced, as has been practiced until maybe the past 20, 30 years, has been one of trial and error, of making inferences based on single patients, on anecdotes. And of course, this method, which is the sort of tried and tested method of classic medicine, is at huge risk for bias and confounding, right? Just because you observe something in one patient doesn't mean that it's true for a population. And so this is what has led, of course, to epidemiology, to evidence-based medicine, which I think has certainly contributed to significantly advancing the way we think about disease and the way we treat disease. But it has many, many pitfalls. It has many risks, right? So one of the risks of evidence-based medicine is that we tend to cluster patients together that maybe are quite different. We have this idea of one size fits all, right? So we're going to treat all patients with, let's say, hypertension in a similar fashion. And you have hypertension, which affects maybe 50% of the population, is this hugely heterogeneous disorder that has many, many biological subsets. So this approach, which has led to standardization, to protocols, to algorithms, to guidelines, this is really the way we practice medicine in 2017. I would argue that this approach, which is hugely valuable, is really reaching its limitations because we realize that one size does not fit all, and in many cases, such as in TBI, it hasn't worked, right? We don't have a treatment for TBI. We're not really good at predicting TBI outcomes. And so this is what has led to newer approaches, thinking about not just the phenotype or some gross imaging marker, but thinking in terms of the entire realm of data that is available on a single patient, right? So that means looking potentially at gene expression or protein expression. It means obtaining quantitative imaging. It means very detailed phenotyping, perhaps using wearable sensors. And this is, I think, where we're potentially moving in a very short period of time right now. It also means potentially using smart or adaptive clinical trial designs to better select those patients that we think are most likely to respond to an intervention. And the hope is that by applying these methodologies in populations, we could then, this will trickle down to an approach where we could characterize the biological specifics of individual patients and using that information potentially tailor therapy, individualized therapy for care. So I hope this makes some sense, but I'm going to illustrate this a little bit more. So the question then you could ask is, do we really need precision medicine? Precision medicine? I would argue emphatically that yes, we do, because current phenotype-based descriptions, clinical descriptions that we make at the bedside are good, but they're not great. They're not great in terms of really telling us about the underlying biology of our patients. They lack specificity, and the same applies for treatments, right? The treatments that we implement in many of our patients are nonspecific, and they have significant toxicities. And we're not very good at prognostication. And so this is the sort of the basic idea of precision medicine. It's to integrate data at multiple scales, going from the molecular to the entire organism, to the actual environment, social and physical environments of our patients. And thinking globally about these different layers of data, potentially we can arrive at a more precise idea of how a single patient is doing and how that patient will respond to treatment. So what is driving, yes?
So this makes a lot of sense. Maybe by the integration of data across different levels, we will understand other things. But just by doing that, it's a little bit shooting on the dark, because you also have to inform these by, like you said, biological processes that maybe. So not just by integrating data, we come out with solutions of things. The pathophysiology of the problem, because we couldn't see before because there was no integration.
Yeah,
certain situations may happen, but others may not.
I think you're touching upon a fundamental question which also arises in regards to precision medicine, which is that we're talking not only about a potential shift in the way that we might think about disease or practice medicine, but we're talking also about a shift in the way that we might conduct research on disease, because the classical approach, of course, is to formulate a question, a hypothesis, a model, and then work your way through, either animal experimentation or in humans to try to demonstrate whether or not your hypothesis or your model is correct. And in many ways, what precision medicine is proposing is a very different, non-model, model-free approach, where you would essentially collect data, and that by applying specific statistical techniques, computational approaches, artificial intelligence, machine learning approaches to these complex data sets, that actually some significance will emerge, that hypotheses might emerge from that approach. But I think that there is a risk, of course, that in many cases, we can go on a fishing expedition and we can pour through huge amounts of and many different layers of data without arriving at anything that is significant. So I think what we need ultimately is a good balance between hypothesis, model-driven research, and data-driven research. And I'm not sure if I'm answering your question completely, but one question that also arises is, why now? Why are we talking about precision medicine now? I mean, we've been doing genomics for more than several decades. We've been doing protein expression arrays. We've been doing advanced imaging for many, many years. Why is everybody so excited about precision medicine right now? And I think one of the big reasons is economic. It's financial. And the fact is that you all are familiar with the Moore's Law, which essentially states that the cost of computing is going down over time, and that the power of computing is going up in an almost reciprocal fashion. And yet, when you look at the cost of sequencing the genome, it actually is going down, but at a rate that is even faster than would be predicted by Moore's Law. So probably part of it has to do with better computing, but part of it may also have to do with more sophisticated technologies in terms of molecular sequencing. And I wanted to show you this. This is a paper from the New England Journal of Medicine, where a review on precision medicine. But I want you to look at this blue line, okay? This blue line represents a sort of hypothetical cost of phenotyping. It represents, for example, what it would cost if you have a patient, who is admitted to the hospital for evaluation of congestive heart failure. You bring this patient into the medicine service, and you do, an EKG and a chest X-ray. You examine this patient. Maybe you get an echo. Maybe you get a cardiac cath. And then a day or two later, you release this patient after modifying their treatment. But what I'm trying to show you is that it's quite clear that the cost of sequencing the genome is now potentially has reached this inflection point where it's probably much cheaper. In fact, it is. It costs about $600 to sequence your genome. To admit a patient with heart failure for 1 or 2 days is, I don't know, maybe $10,000, $15,000. So it's cheaper today to actually sequence all the information that is really kind of the cellular hub of all the information that is going to happen in your body than it is to perform a routine sort of history and physical and get a few additional lab tests. And so why is this important? It's important because it means that it's becoming economically feasible, actually, to perform precision medicine in large numbers of patients. And this is what is behind this concept of advancing today with precision medicine. The basic idea is that we collect data. We collect data at multiple scales. We collect detailed quantitative data. This generates information. Then using computational approaches and other statistical methods, we potentially arrive at some new knowledge or some new insights on disease. Potentially this will lead to modifications in health outcomes. And the idea is that these changes in health outcomes will then inform the data stream at the origin and will help us in a sort of recursive or iterative way to potentially advance science and biology. And I think that this idea is very powerful, and it's really what has been behind the precision medicine initiative. Doesn't this seem kind of old? It seems like it was ages ago that he was talking about it, and yet it's been only a few weeks. But anyway, so our dearly regretted, former President Barack Obama actually was a big proponent of precision medicine. Even in his days as a senator back in the early 2000 period, he was advocating for precision medicine as a new approach to thinking about disease. And during his tenure, he launched this initiative 2 years ago, which is essentially is an alliance between the NIH and the DoD, the DARPA, to enroll 1 million individuals, healthy individuals, ill individuals across the country, and perform very, very detailed phenotyping, imaging, genome expression analysis, proteome, and sensor-based measurements, and follow these people over time, right? So this precision medicine initiative is actually underway. There are a number of centers, not Johns Hopkins, but a number of centers have been funded to carry out this enrollment and this research. And the expectation is that this huge data collection effort, 1 million individuals, is going to potentially transform the way that we think not only about disease, but also about health, and how can we potentially reform our ideas about classification. So let me talk a little bit about, in the remaining time that I have, about traumatic brain injury. And I've kind of put this down as 6 tasks, 6 Herculean tasks to potentially transform the way that we deal with traumatic brain injury patients. I think first we do need to understand more about the biology of TBI. TBI is a very complex disorder, and it's notwithstanding a great deal of interesting basic science and translational science in this field, we really haven't arrived at a sort of unifying hypothesis about how TBI occurs, how it damages structures in the brain, and how over time it can result in different types of deficits, but also can result in regeneration and repair. I would like to talk a little bit about imaging, because this is an area that I do my research in primarily. We'll talk about the electronic health records, biosensors, clinical trials, and knowledge networks. So I think perhaps the most important point is that we don't know enough about the biology of TBI. TBI is a very complex disorder which affects not only neurons but glial cells. It affects the function of synapses. It disrupts neural circuits at the microcircuit level and also at the network level. And it also disrupts the vasculature, both the macrovasculature and the microvasculature. So it's a very complex and heterogeneous disorder that affects the brain in many different ways, and the specific preponderance of these different mechanisms may vary considerably between individuals, and also depending on the underlying mechanism, whether we're talking about an impact or a blast, or whether we're talking about an acceleration or deceleration type injury. And so another point, just to sort of be emphatic here, is that TBI is really not a localized disease. You should never think about, even though you might see a patient with a focal parenchymal contusion and say, okay, this is just a localized process. It is not. In fact, TBI is a disease really of systems, of circuits. And this has been shown abundantly in a number of studies in animals and humans. One of the ways that we've been able to really understand the fact that TBI is a disease of neuronal systems is that we are able now to map out the white matter, for example, using diffusion tensor imaging. And we can see very clearly that even in the mildest forms, in concussion, patients who apparently have had a very light or mild injury, you can see that the white matter is disrupted. And it's disrupted in a multifocal or disseminated way in almost every case. So this diffuse axonal injury is really one of the hallmarks of TBI of all severities. Yes? Yes, this was done in the acute setting. I think it was like 5 days after TBI. And so, we arrive at an interesting kind of model where we know that the connectivity or the networks of the brain, over time, because of aging, perhaps because of vascular processes, perhaps because of amyloid deposition, this network loses some of its integrity. But what we know as well is that TBI actually provides an interesting scenario where that loss of connectivity occurs much more rapidly. And so what you might see in some individuals, at least, is a cumulative effect of both age-related cognitive impairment due to disconnectivity or loss of connectivity, compounded by the disconnectivity that occurred because of TBI. And so what TBI may do in some cases is it may reduce the cognitive reserve that individuals have that allow them to potentially suffer a brain insult or an encephalopathy without major symptoms. If a patient has had TBI before, it's possible that that amount of reserve is diminished and they will be much more symptomatic. In fact, we have some data to suggest this. This is just a slide demonstrating the very dramatic effects of this chronic traumatic encephalopathy. The brown stains here are actually selective binding of tau protein. You can see that there's significant cortical atrophy that is occurring. This is a 23-year-old individual who committed suicide in the setting of chronic traumatic encephalopathy. And you can see the brain is shriveled up, and there's a very dense deposition of tau protein in the cortical layers. And so the question is, can we understand more about this process through precision medicine? And of course, this is what is familiar to all of you, this basic dogma in biology of genes that are, of course, leading to transcripts that are leading to translation to proteins that then lead to the specification of specific metabolites. And the question is, can we exploit this type of paradigm to better understand what is happening in TBI? And I think the answer is probably yes. What we need to do, though, is maybe to get rid of the very simplistic linear associations that many people have tried to make between specific genes being increase in expression or decrease in expression and specific phenotypes. And what we know is that genes never, and proteins never act in isolation. They're part of networks, right? And so what we really need to understand is how the system of genes is potentially modified in specific individuals, how the system of proteins is modified. And using, again, methods from systems biology, we can develop very, very interesting hypotheses about perturbations of these systems and how these perturbations lead to specific clinical phenotypes. So if we talk about genes and genes that have been associated with traumatic brain injury outcomes, a number of associations have been made, and I'm just showing you some examples. There's many others. But the question is, what is the significance of these associations? It appears that some, single nucleotide polymorphisms, so some variants in gene expression are associated with specific recovery phenotypes after TBI. But the question is, can we actually, using these types of associative descriptions, can we actually arrive at some biological hypotheses about what is driving recovery after TBI? So it's a very interesting, one of the genes that has been shown to be significantly depressed after TBI is DISC1, disrupted in schizophrenia 1. This is a kind of an interesting scaffold protein that really orchestrates the movement of many proteins in the cell and seems to play a hugely important role in two things. One is in the division and differentiation and migration of neuronal stem cells. And the second is in the expression of glutamate receptors at the synapse, and therefore it plays a role in synaptic plasticity. So both a role in neurogenesis and in synaptic plasticity. So this is quite possibly the single gene that you wouldn't want to depress, or after a TBI, you really want this gene to be actually increased in expression to allow potentially some regeneration to occur after this damage. In fact, it turns out that DISC1 is dramatically reduced. Interestingly, this DISC1 has been described by investigators here in Johns Hopkins. The group of Guoli Ming and Hongjun Sung have done some really, really nice work demonstrating how this gene, mutations in this DISC1 gene, which have been associated with severe mental disorders like schizophrenia and depression, actually you can, by deriving somatic cells from individuals who have this mutation and de-differentiating them into induced pluripotent stem cells, you can then have a model system where you can induce a neuronal fate, and you can observe in vitro the synaptic characteristics of these cells. And this has led to some very, very interesting hypotheses about how DISC1 plays a role in determining aberrant or abnormal synaptic function very, very early on, possibly during childhood, in individuals who are at risk for schizophrenia and for depression. But I guess the point I'm trying to make here is that it's possible that this DISC1 plays a very important role in determining post-injury, for example, post-stroke or post-TBI, regeneration or inhibition of regeneration, and this is something that we're beginning to look at in our lab. There's also quite obvious evidence of epigenetic changes. So epigenetic changes meaning those changes that do not involve the actual sequence of base pairs in the DNA, but involve, for example, modifications in histones or methylation, acetylation, that occur after TBI, and it's likely that these modifications play a very important role in determining gene expression and protein expression, and we know that they are quite important and dramatic after TBI. Another form of epigenetic change is the expression of these microRNAs that can be actually released from cells and can be measured in the extracellular fluid and also in the bloodstream. And increasingly now, these microRNAs are being recognized as potential biomarkers of neuronal damage as it occurs in TBI, but also providing clues as to the underlying neuronal changes that occur, both in terms of the injury and also in terms of the regeneration. And this is another example of the molecular analysis that can be applied. This is a lipidomic analysis, again, in TBI, looking at expression of different phospholipids in an experimental model of TBI. This is some of our own data, and this is, was done with my colleague Akhilesh Pandey from the Proteomics Discovery Center. We have looked at a small number, 25 patients with concussion. We took their blood, and we've been analyzing lipid expression, and we found that selected lipids seem to be increased in expression in concussion subjects compared to healthy controls. And the question is, what is the significance of these lipid changes in the blood, and can they inform us not only as biomarkers to detect injury, but could they potentially help us understand something about the biology of neuronal damage? And then this is some very interesting work that we've been doing in my lab. This is actually not TBI. It's a mouse stroke model, but we've been interested in the, like many others, in the composition of the microbiome in the gut of these mice. What we did was we gave some of the mice antibiotics, and some of them we gave them no antibiotics, and then we gave them all a stroke. And we found that, so when you give antibiotics, and this is just essentially a clustergram of all the different types of microbes or bacteria that are found in the gut of these mice. And you can see that when you give antibiotics, you deplete, of course, the bacteria. But more interestingly, we found that when you give antibiotics before the stroke to mice, you actually decrease the size of the infarct quite significantly. And And we're just trying to work out what the significance of this. But I bring this to your attention because it's likely that alterations in the microbiome, not only in the gut, potentially in the skin, potentially in the lungs, could have a major effect in terms of modulating immunological responses and determining the degree of recovery that occurs after TBI. So just a few words about mapping the brain. It's very clear that non-quantitative, qualitative approaches, CT scan, for example, does not allow us to discriminate. So these are two of my patients in the neurocritical care unit. They both had identical clinical characteristics. They were both identical in terms of their CT scan, or almost identical. One of them, this patient actually survived and had a great functional outcome. This patient died. And yet, at the outset, these patients were indistinguishable. So can we do better? And the idea that we have and others is that maybe since we postulate that TBI is this disease of neuronal systems, of circuits, maybe we can interrogate those systems in vivo using advanced MRI. So the technique that we've been using over the past 3-4 years is diffusion tensor imaging, and this is an example of tractography, combining that with resting functional MRI to look at how different parts of the brain are activated at rest and determining the functional coherence between these different regions. So this is just an example of how TBI, this is a control subject, and you can see this kind of spaghetti of white matter tracks that is quite robust. And here, by comparison, is a subject who had a TBI 6 months earlier, a severe TBI. And you can see this rarefaction of the white matter tracks as reconstructed here in 3 dimensions. So very, very dramatic effect. And this is what I was talking to you about earlier, which is how the fact that TBI is this multifocal, diffuse disease involving many, many different regions of the brain, and in particular the white matter. So we did this study where we looked at the integrity of white matter tracts in 100 patients with severe TBI, and then we followed the survivors over 1 year, and we got their functional outcome scores. And what we found was that when we compared the impact score, which is what I showed you earlier, it's the CT and clinical classification, when we compared that with a new score that incorporated CT, clinical, but also a composite DTI image of the white matter, we were able to increase discrimination significantly. So the area of the receiver operating characteristic curve went from about 0.6 to 0.9, right? So this is practically a doubling of our discrimination. And this was a very impressive result. And since then, we've done some additional studies. This was a study where we looked at a very large number, 10,000 patients who had undergone functional MRI after brain injury. And we're trying to determine which were those regions of the brain where the signal was most profoundly depressed in comatose patients. And we found that the area that seemed to correspond best was an area that involved the thalamus, bilateral retrosplenial and precuneus, and also some medial frontal structures. And these are structures that form part of a network which is called the default mode network, which is postulated to be significantly associated with loss of consciousness, and as it is also associated with many other functions such as introspection. And so based on these preliminary observations, we conducted a study of patients with severe TBI. This was in 28 patients with severe TBI who were admitted to the NCCU. We took them to the MRI scanner, and we did diffusion tensor imaging, but we also did resting functional MRI to identify the functional architecture of these severely injured patients. And what we found was that when we focused on specific networks, this is that default mode that I was mentioning to you earlier. This is the attention network. This is executive control network. And this is a salience network, which is involved with identifying salient features in our environment. When we compared control subjects with those TBI subjects who had a favorable or unfavorable outcome, there were significant differences. But then again, there seemed to be major differences between those who had a favorable and those who had an unfavorable outcome in terms of their connectivity. And the most dramatic effect was actually not so much the coherence or connectivity within specific systems or networks, but the disconnectivity that occurred between networks. So, for example, the anti-correlation that is normally seen between the default mode and the salience network, which is something that is characteristic of all normal individuals. When you open your eyes and engage in a task, your default mode tends to shut down, your salience network tends to light up. That anti-correlation actually is lost in patients with TBI, and in particular in those patients with TBI who had a poor outcome. So what we think is that maybe this represents an interesting kind of biomarker of the types of functional changes that might result from this massive damage that is occurring to the white matter tracts in the brain. And there is some biological plausibility to this. This is actually a very, very interesting study that was published looking at these different networks that I described to you, default mode, salience, and also determining the gene expression. So they had access to autopsy specimens in a subset of patients, and they looked at gene expression, and they found that there was correlated gene expression that seemed to correspond almost exactly to these functional maps of networks that we are obtaining using resting functional MRI. So there is some sort of, we're not just talking about blobs in the brain, we're talking potentially about something that could be biologically plausible. And there is, of course, increasing focus now on resting as opposed to task-based fMRI techniques to assess the integrity of the functional architecture in the brain. But just to tell you that there are so many other ways that we can map the brain. This is mapping of amyloid, and we know that amyloid, of course, is associated most significantly with Alzheimer's dementia. But it turns out that in the setting of TBI, amyloid deposition is dramatically increased, and this amyloid deposition never quite goes away. So this is a subject at 48 hours, 54 days, and nearly 1 year after TBI. And you can see it's moderate and severe TBI. This patient, you can see the significant amyloid deposition, which is present, which is totally abnormal. This is a 34-year-old subject. So something that might suggest a potential link between, again, the acute setting where you have an insult, this diffuse axonal damage, and these long-term sequelae that we're observing in many patients with TBI. This is another PET-based radioligand that is selective not for amyloid, but for tau protein. And it's been developed more recently, and you can see these are retired NFL players. And you can see that when you compare controls, those subjects who had sustained TBI during their tenure in the NFL had significantly increased tau uptake using this technique, and the uptake was actually comparable to what was seen in subjects with Alzheimer's disease. And this is data from Hopkins with a third type of radioligand. This one is for TSPO. So the TSPO is essentially expressed in activated microglia, so it's used as a marker or a surrogate for inflammation. And you can see that in this particular former NFL player, years after he had sustained TBI, there was still evidence of significant microglial activation throughout the brain. I should say that this TSPO is not specific for microglia. It also binds activated astrocytes. So just a few words about biosensors, because I know we're running short on time, but I think this is something we can't overestimate the importance of wearable Wi-Fi-enabled biosensors. There's an increasing focus now on sensors that can measure not just motion or motor outputs, but also the composition of the chemistry in your skin. It can measure as well the different electrical circuits that are generated at the surface of the skin. So I've been working for the past year, actually, with a group from the Department of Biomedical Engineering in Homewood to develop a multi-segment sensor system that allows us to essentially measure and track in real time the motor outputs of subjects. And so we're currently using this system to classify different types of movements that we see, for example, in coma patients in the NCCU. We've obtained a high degree of accuracy in classification with healthy subjects, and now we're going to implement this in the ICU to determine whether we can, in a sort of automated, unbiased fashion, obtain classifications of motor outputs. And then, of course, this is going to lead to a number of other interesting projects, but just to sort of give you a sense that this is also what we're talking about when we talk about precision medicine. In terms of clinical trials, well, it's quite clear that, in the realm of oncology and cancer, the approach to treating cancers has really been revolutionized by the identification of specific molecular targets. And we know that the outcomes in many types of cancer are dramatically different now than they were even 10 or 15 years ago. And I'm thinking, for example, of melanoma or breast cancer. So the question is, can we apply similar methodologies or approaches to think about treatments for conditions like TBI? And here we need to talk about two different types of enrichment. When we talk about enrichment, we're essentially talking about selecting a subpopulation within a specific disease category. So prognostic enrichment essentially means identifying those individuals that we think are most likely to encounter a specific outcome. And this can be based, again, on this multi-layer data set combined with clinical and imaging characteristics. And this has to be contrasted with predictive enrichment, which is really a clinical trial strategy. It means essentially to identify those individuals using specific markers, to identify those individuals that we think are most likely to respond to a specific treatment, and enrolling preferentially those patients into our clinical trials to maximize the likelihood of demonstrating a treatment effect. And even though this seems very simple, it's interesting that the vast majority of clinical trials that are being done in medicine today are not using these enrichment strategies. It's clear that we do need to do this. And a major emphasis in clinical trials moving forward is going to be to use biomarkers, right? Either biomarkers obtained at the outset to select patients for specific treatments, or biomarkers that are obtained along the way after a specific treatment, get the biomarkers, and then continue to treat only those who are positive or negative with the biomarker. So this adaptive clinical trial design is something which I think is hugely important. In the last few minutes of this talk, I'd like to just briefly mention to you that right now there are a number of ongoing data collection efforts that are hugely important in the realm of TBI. So one of them is just the basis for many of these efforts is what's called the Common Data Elements, which essentially is a group of experts that met in the NIH to determine specific clinical research imaging definitions for specific endpoints with respect to TBI. And then based on this Common Data Elements project, which really was put into place about 7-8 years ago, a number of studies have evolved. There's the TRACK TBI, which is an NIH-sponsored multi-center effort. The target enrollment is 3,300 patients. And the goal is essentially to recruit patients with TBI across the severity spectrum and obtain very detailed imaging, phenotype, genome, and proteome, and in a subset of patients to follow them over time to determine how we can build better prediction models. There's a very similar effort that is being run in Europe. It's the Center TBI project. Now the target enrollment is 5,000. And this effort is ongoing, and I have the privilege of being a part of both of these. For pediatric patients, there's this Approaches and Decisions in Acute Pediatric TBI, ADAPT trial. There's a very interesting effort, the Chronic Effects of Neurotrauma Consortium, which is led out of VCU. And then there's the Concussion Assessment Research and Education. All of these are accessible online, and all of these efforts, I'm pointing them out to you because all of these efforts are funded federally, which means that we, as being part of the scientific community, have potentially access to these data sets. In fact, I can tell you that, for example, for the TRACK TBI, it's not very complicated to make a request and obtain access to very detailed data sets in TBI patients. And this could be a huge advance for people, especially who are beginning their research careers and who don't have necessarily the means to institute on their own their multi-center trial. So there's also these data sharing platforms. There's if you look it up online, the FITBEER, which is the Federal Interagency Traumatic Brain Injury Research Consortium, which essentially is a clearinghouse or a platform that allows people with TBI data, whether it's experimental or human, to deposit their data sets into this repository, and this data can then be accessible to the scientific community. And there's a similar effort that is being done internationally, the INTBEER. I just wanted to also point out that these two large-scale initiatives, the BRAIN initiative, which was launched maybe 4 or 5 years ago, initially called the BRAIN Map, and then the Human Brain Project, so the BRAIN initiative here in the US, funded by NIH, and the Human Brain Map, funded by the European Union. These are huge opportunities in terms of funding, and I think that I'm bringing them up because they also represent opportunities to use TBI as a model system to interrogate the neuronal architectures and circuits in the brain. And while a lot of this is focused on basic science and potentially non-disease-related neuroscience, in fact, there are opportunities to tap into these funding streams for TBI-related research, and I can point you, if you're interested, to specific projects that are doing this. As far as precision medicine is concerned here in Johns Hopkins, I think you're familiar with the InHealth, or Individualized Health Initiative, which is run by Scott Ziegler and Anthony Rosen. And essentially what it is an alliance between the health system, the Applied Physics Lab, and the university to really push the agenda forward in terms of precision medicine, both in the realm of biology and also in medicine. And this initiative, which currently is still trying to find its way and is not sufficiently funded, it's likely that this will become a much bigger deal, and that we're going to see in the near future the emergence of a true precision medicine institute as part of the Johns Hopkins system. So in conclusion, I just wanted to sort of give you a flavor of TBI. TBI is, I hope you've been able to appreciate, is a major public health concern. We really don't have any accurate markers for detection, for prediction, for treatment selection, and there's no effective therapies, right? So the opportunity here, I think, is precision medicine, and here we're talking about capturing the biological sort of signatures of individual, TBI patients or subgroups using the methods that I described to you, and of course working in a collaborative fashion with our colleagues in engineering and statistics to develop the appropriate models that can help us understand these complex data sets. And of course, getting engagement from investors and stakeholders, because while this is becoming a very, very important topic right now, this was not the case before. And perhaps one of the reasons, again, it's economic. The cost of sequencing the genome in the year 2000, was about $15 million. And as I mentioned to you today, it costs about $400 or $500 to sequence an individual's genome. So here's kind of like the sort of roadmap. We have individuals that arrive in the hospital with an acute illness, surgery, injury. They undergo these trajectories in the hospital, and they have long-term outcome trajectories. And of course, one of the tasks for precision medicine is to generate better biomarkers, again, for detection and classification, but more importantly for prevention and for treatment. So I'd like to acknowledge support from these different institutions, and also recognize a lot of my collaborators that we're currently working with, both here at Johns Hopkins, Department of Radiology, Department of Neurology, Department of Biostatistics, proteomics, and then also a number of colleagues in other institutions in Europe and also in the US. So with that, I will stop, and I'm happy to take any questions. Forgive me if I had to rush through that a little bit. But, yes?
You mentioned antibiotics in part size and the gut bacteria. Is there any indication that going the other way and giving things like probiotics will have any positive effects in that?
Yeah, in fact, that's it's a specific hypothesis that is being tested by one of my colleagues in Berlin, and so far the results have been not very conclusive. But there is a great deal of interest in this kind of brain-gut axis, as you probably know. And on the one hand, it seems like an acute brain insult, like a stroke, can have profound effects on, for example, gut permeability, translocation of bacteria. On the other hand, it seems like modifications in the flora of the gut can profoundly modulate the immune system and lead to changes in the way that the brain responds to an injury. And so the question is, can we interfere in that cycle, potentially by giving antibiotics or by probiotics? I should mention, there was a large randomized controlled trial, I think it was published just a few months ago in Lancet, looking at the effect of post-stroke antibiotics in humans. And this was done in Europe. Unfortunately, it didn't show any benefit. So I think there's still a lot of things that we need to learn. But the point is that the microbiome represents potentially a very, very important opportunity to modulate the way that the brain is responding to an injury. Yes?
I think I have a hard time understanding the concept of integrating all the data together, almost to the point where it could be too much information, because with any small changes, it could be so multifactorial, like a lot of compounding factors. And adding more information, how can that help us kind of pointing out at this point, this is probably this micro MRI change is causing this, or you know what I mean? Is it so many steps in between? There's time variables. There's
So,
information. So how do you process the whole data?
So I would respond by saying that you're not alone. I also have numerous difficulties trying to think about the enormity of the task. I think this is probably the most important question, because it's, in a way, you could say that it's not so hard to collect data. It's not so hard to obtain imaging, to obtain, to sequence the genome. But making sense of that in a way that is relevant clinically and can change the outcome of our patients is a much more difficult task. So that analytical task and the construction of appropriate models is really where the heart of this problem lies. And I don't think that anybody has the answer. So believe me, you're not the only one who feels that this is a daunting task. Yes?
So in rehab, we're all aware that one of the strongest predictors of outcome is something that, at least I'm not seeing in non-old medicine, is psychosocial factors in the environment. And I'm wondering how you're able to factor that in. Looking at some of the genetic or hemodynamic indicators.
Yeah, I think most theoreticians of this precision medicine process believe that the environment, including the psychosocial context, is part of that multi-layer data set. So that means that you can't pretend to be comprehensively collecting data and analyzing your patients without actually integrating that type of information. So I guess I would answer by saying that is part of the precision medicine construct. It's sometimes difficult, I guess, because, how do you quantify a lot of these things? How do you quantify, for example, the richness of somebody's social network? How do you, and really what precision medicine is about in many ways is about moving away from qualitative approaches towards more measurable quantitative approaches to all these different data sets, right? And so, so I would challenge people to think about, what are useful metrics that we can use to integrate things like the psychosocial environment into precision medicine models. And I think that's something that, we should do.
But going on that, there are people who do research in science on psychosocial and so on. They have quantitative metrics, things you can argue whether how sensitive they are, how precise they are, and so on, but they use quantitative metrics for psychosocial. And I think that the behavior is a contribution that the rehabilitation specialties can do and contribute to the spectrum of depressive medicine. So that's why I don't want to play that. That's important, and it used to be paired with other elements of data that you can potentially expect.
Yes.
One comment and then a question. So I think just to elaborate on Dr. Mayer and Dr. Selnick's point, that you had that slide about TBI and veterans and dementia outcome. For example, was rank a covariate in that study?
Rank,
that would give you an idea of their their socioeconomic status, for example, and their risk of being exposed to.
Yeah, I seem to recall that they did a multivariable model and they adjusted for different things. I'm not sure if they adjusted for rank, but they found that exposure to TBI was independently associated with a higher risk of dementia. And that graph that I showed you was just one out of many presented. But, sir, you had a question?
I think one of the points that when I think about, I need to think about the definition of precision medicine a little bit broader.
Yes.
And that is, the purpose of precision medicine is to try to identify some characteristic about a person that suggests their responsiveness or prognosis as well as their responsiveness to treatment. The current thinking about precision medicine is that the genome is the place to look for that. And I would argue that it will take some time for that to bear some fruit. And that it would be useful to, but the concept, though, is very useful for all of us. That is trying to identify unique characteristics of this person, their genome, their social situation, their cognitive status, their other statuses that may indicate their responsiveness or appropriateness to certain kinds of treatment. So I think the model is a very powerful model, but it's not just the genome to look at. There are other classes of variables to look at, but the model is very, very important. And I think it's easy to say, well, you know, the genome does not relate to rehab, but I think the model is very relevant to rehab, although we may be looking at other kinds of factors that determine outcome. And in many ways, precision medicine, in the broadest sense of precision, is the future of medicine.
I couldn't agree more with you, and I think at the beginning of my talk I did emphasize that, you know, the genome is just one layer out of many different layers of data that we need to try to think about in developing this individual signature of people, and it relates not just to disease, but it relates to health as well. So I agree completely with you.
I guess there's one other thing. Dr. Stevens' presentation of these various databases, I think, is very, very useful for us to be thinking about in our department, because people think, oh, I can't get a big grant to look at data. Well, in fact, there are large databases out there that can be queried with appropriate permissions and support. And I would just add to the ones that he did, of course, the Model Systems TBI database is the largest brain database in the world. 14,755 people as of yesterday, well over 25 years. Nothing is remotely, as far as I know, near that size, although it's older, so some of the data is not so good. But that also can be queried for free. And I think our residents and a lot of our faculty should be looking to these large data sets to begin to investigate questions, particularly when funding is so tight. And there are important questions to be answered.
Okay, well, thank you very much.